You're listening to Making Waves, fresh ideas in freshwater science. Making Waves is a bi-monthly podcast where we discuss new ideas in freshwater science and why they matter to you. Making Waves is brought to you with support by the Society for Freshwater Science, Arizona State University's School of Life Sciences, and the University of Washington School of Aquatic and Fisheries Sciences. Welcome to the Making Waves podcast. I'm here this month with Dr. Amy Rosemond, who's an associate professor at the Odum School of Ecology at the University of Georgia in Athens. Thanks for joining me, Amy.
Thanks for having me, Eric.
And, well, you've been doing great work for a long time, but a recent paper of yours was just published in the journal Science, which is among the top journals in our field. And so I wanted to talk to you about some of the research results from that project. And so your paper discusses how nutrient pollution may affect the carbon storage of streams, particularly in regard to detritus. And I first wanted to ask you, just why do you think it matters if streams are, as a whole, losing or sequestering carbon?
I think it can be viewed from both a food web and a global carbon cycle perspective. We understand now how important detrital carbon is for the health of streams, for production of organisms, and the uptake of pollutants. If only algal resources are considered in a nutrient pollution scenario, you'd think, well, that's a good thing. It's going to stimulate algal productivity and sequester some carbon, even if that's even short-term sequestration. But as all of us know, streams, they contain a much greater proportion of detrital versus algal carbon as their resource base. Mm-hmm. So, all this terrestrial carbon is entering streams, and the fate of that really matters. Is it respired as CO2? Is it transported downstream, or is it retained there for an amount of time that gives rise to things like production of organisms and uptake of pollutants? So when we found that nutrients accelerate that carbon loss, that means that there's less carbon there, less production, and less uptake of pollutants.
Right. And so one of the main points that you made in your paper was that we sort of had a much better understanding of how nutrient additions will affect these algal-related carbon processes, but not detrital-related processes. Is there a reason why you think that is?
I guess because we can visualize algae and its greenness. Mm-hmm. You start realizing how limited we are as humans in that we tend to respond to things that we really can see. When nutrients stimulate algal productivity, we see things get green and slimy. Yeah. And it's just not as apparent if we are losing detrital carbon from an ecosystem. It's just hard or impossible to visualize that.
I don't know. Nowadays, when I look into a stream, I see all kinds of leaves everywhere, but maybe to the average person, that's not the case.
Go back in a month and another month and another month, and then you can determine some rate of loss there.
So one of the things that your research suggested was that the presence and perhaps even the type of detritivores might influence how this carbon loss occurs. So could you talk a little bit about what types of detritivores are living in, at least in the streams that you were working in, and why this might be important?
That's sort of the link to the rest of the food web, right? But some streams have more shredders and some have fewer. And so you can think of this carbon on the bottom as having different pathways to any top consumers like fish, and then other functions that it's playing in terms of just supporting uptake of pollutants and other functions. And so in thinking about the sort of three alternative fates of that carbon, it's retained in the stream, or it's being lost as CO2, which is primarily a microbial-driven process, or it's being transported downstream into the next reach and respired there. Mm. Detritivores are going to accelerate that loss from the reach and converting large particles of detritus into small particles that influence its transport then in the whole river network. In an agricultural stream, a lot of high nutrient streams might have other stressors that could suppress microbial activity. They could suppress detritivore activity or abundance. And in streams where there's a lot of nutrients, but there's not as many detritivores, you can imagine that more of that carbon loss is going to be via CO2 loss versus the transport downstream.
If it's lost as CO2, then would it be less available to the food web?
Right. You can imagine that that's the ultimate loss as it goes through the whole river network. And so if all of that was dominated by microbial loss processes, it might actually result in a greater net carbon loss from the whole river network than when you had detritivores present.
Much of the research that you've done, including the work that we've been discussing, has been conducted at a site funded by the National Science Foundation Long-Term Ecological Research, or LTER program. And I was wondering how having this sort of long-term data and the logistical support associated with this program help you accomplish a study that is so large in magnitude as this one?
It took a few years, huh? But I think I've gained so much from, my collaborators and the infrastructure. And so the ideas, the models, the context of the data that you're collecting, to have a collaborator like Bruce Wallace, who pioneered whole stream ecological research at Coweta, and whose work built on things that were being done at Hubbard Brook and all the knowledge and approaches from the U.S. Forest Service, who started doing these watershed manipulations back in the 30s at Coweta. And there's all the data from the many investigators that have worked at Coweta that gives then grounding and context to the findings that you have from that same site. In terms of logistical support, I'd have to say that some of the most critical was when we almost went off the road. we were selecting sites, and it was early January, and there had been snow and ice. And there's an area at Coweta called Devil's Ball Alley Cliff. Mm. And anyway, the truck we were in, some of the back tires slid on the ice, and we had to call down below. Luckily, someone got cell phone reception up there. And anyway, getting bailed out was a really great advantage of being part of that Forest Service and LTR infrastructure.
Yeah, so getting back to that, this paper seemed like it involved a ton of work. How did you conceive of such a major undertaking?
I guess it was, like most things, one step at a time. For my postdoc work, I had worked down at La Selva with Kathy Pringle and Judy Meyer as my mentors, and was interested in a phosphorus gradient and what effects did it have on these detrital processes. Going from that landscape gradient, I then collaborated with Bruce Wallace, Keller Supercrop, Pat Mulholland, to write our first whole stream nutrient enrichment proposal. And so we started with one stream compared to a reference for several years. Then the next step was to ask questions about nitrogen versus phosphorus control of these processes. And then with a different team, now including John Benstead, Vlad Gullis, and John Mayers, we're proposing to enrich streams, multiple streams, with different ratios of nitrogen to phosphorus over a 3-year period. The data that's in the science paper was sort of a eureka moment of thinking about how we might use the information from all these studies to look at this process of whole stream rates of carbon loss.
You've been interested in nutrient addition types of studies for a long time, but what made you first become interested in detrital processes? As you mentioned, they're sort of harder to see and perhaps less evident, but why do you think they're so important, and how did you get interested in that?
My dissertation work was all about top-down, bottom-up control of stream periphyton assemblages and productivity. And I guess anyone who is working in stream ecology in the last 2 decades, and starting, let's say, with Hubbard Brook and Coweta, and just realizing that so much of what is fundamental to streams is that detrital resource base. So it was a natural transition for me. I guess I'd also done some experiments as part of my dissertation that I never published, where I started to play around with nutrient effects on litter breakdown. And of course, the work that was done, Walker Branch, was where I had conducted my dissertation work. And so it was work as early as the 80s there that had also addressed some detrital processes in terms of in relation to nutrient enrichment. So it was really a function of all the things I was exposed to, and just having this view coming out of testing these top-down, bottom-up questions in stream ecology, and realizing that fewer questions were being asked about top-down and bottom-up control in detrital-based pathways.
Well, it seems like with the work that you're doing and that Bruce Wallace has done and a number of other folks have done, now we're really realizing how important these pathways are. And I think it seems like you're transforming the attitudes of people in thinking about this.
These are going to be more or less important in different kinds of systems. And certainly there's a lot of evidence that even a little bit of algae goes a long way in terms of stream function and food web dynamics. At the same time, when we're talking about the whole resource base of a stream, that detritus is going to play an important role in just about any kind of aquatic ecosystem.
So what do you see as the future for your project going forward? Are you continuing these nutrient additions?
We had to turn off the juice. As you can imagine, that takes a whole lot of care and attention and multiple trips to Coweta and to keep all that infrastructure and a continuous enrichment going. So what we have developed is an understanding of the mechanisms that nitrogen and phosphorus have in affecting carbon breakdown, and also how that plays out across the whole food web, from fungal production, where Vlad Gulas and students were working, all the way up to salamander growth and production with graduate student Philip Bumpers and John Mayers. So we're looking, all the way through the food web in a detrital-based food web, and developing our understanding of how nitrogen and phosphorus play relatively different roles from the top to the bottom of the food web. I'm also interested in how other stressors are affecting fates and rates of detrital processing. John Kamenowski and I wrote a review article that came out a couple years ago. Someone was kidding me and saying we should have titled it "Save the Detritus," making the case that these resources are so important in streams and all kinds of global change is affecting their availability and fates in streams. Everything from watershed deforestation to altered hydrology to altered biodiversity and vegetation to things like nutrients and temperature. And I think that both these rates of processing and then the ultimate fates are things that we're interested in addressing.
So could you talk a little bit about the management implications of this research?
The US EPA stated in the year 2000 that the states had to start establishing nutrient criteria. So the states are now tasked with this, and some are doing a better job than others at coming up with criteria for lakes, streams, and coastal areas.
And what are these criteria for exactly?
Excess nutrients are one of the leading causes of impairment and poor biotic condition in streams in the U.S. And it's a difficult problem to address, because you have this non-point source pollutant, and how do you start establishing how much is too much, and then, what are the limits to inputs to a particular system? So it's a huge problem that we need to start to get a handle on So one of the first questions is, what criteria do you set for nitrogen and phosphorus concentrations? Do you use concentrations? Do you use some biological response variable? If a biological response variable is being used, it's typically an algal response variable. But you can imagine in streams that can be heavily shaded, can experience scouring floods, that algae doesn't hold up very well as a bioindicator of nutrients. And because that detritus is an important resource base, that possibly that should at least also be considered as a biological endpoint for establishing nutrient criteria.
All right. Well, thanks a lot for joining me for this interview today. It was great talking with you.
Well, thanks.
You've been listening to the Making Waves podcast, brought to you with support by the Society for Freshwater Science. For more information on this speaker, the Making Waves podcast, or the Society in general, please visit us on the web at the Society for Freshwater Science webpage. Tune in next time for another fresh idea in freshwater science.